Hi, everyone, and welcome to another Project Blue live chat. Today, we are joined by Dr. Clarissa Anderson from Scripps Institution of Oceanography. If this is your first time tuning in to one of our events, welcome, and I'm glad you could join us. If you have any questions during the event, we will be doing Q&A following her presentation, so please feel free to ask any questions you might have in the Q&A function or the chat below. Our guest today, Dr. Clarissa Anderson, is a biological oceanographer with expertise in ecological forecasting and remote sensing. Currently, she is at UC San Diego's Scripps Institution of Oceanography, where she serves as executive director of the Southern California Coastal Ocean Observing System, which is a regional observing system that works to collect, integrate, and deliver coastal and ocean observations in order to improve safety, enhance the economy, and protect the environment. She's been up and down the coast of California for school and work, and now she's here talking with us. Dr. Anderson, thank you again for joining us. The floor is yours.
Thank you very much for that lovely introduction. I'm going to share my screen now, put my readers on because I'm an old lady. Let's see here, and present what could end up being a pretty long talk. So bear with me, and I will definitely have time for questions at the end, but feel free to interrupt too. If you feel excited about a particular thing that I'm talking about, jump right in. Hard to find a word edgewise with me. I do talk fast. Some of you on the line who know me, like Kip, hello, so good to see you. You know how quickly I can go through things. So I will try to calm myself. This is my team, or at least a part of the team that we call SCOOS. So we sit here down at Scripps, as was already mentioned, and we really do represent, though, beyond the people on this screen, we represent a really large number of scientists, partners, all throughout Southern California. So the work you'll be seeing as I go through these slides, and it's just a smattering of what we do, could not be done without all these partners. We only manage to do what we do because there's a lot of leveraging, a lot of partnership that goes on. So I will just advance quickly through these slides. There we go. So just a little bit of high level backup for all of you who are not familiar with what SCOOS is or what that refers to. We could easily be confused with, say, a federal governmental agency, but what we are really is an enterprise of regional groups all around the country. There's 11 of us that do ocean observing at this regional scale around the country. We are funded by the National Oceanic and Atmospheric Administration, but that doesn't mean that we are feds, right? We get money from that group to do 5-year chunks of activities, and we try to maintain our portfolio around a certain mission and vision that is a collective vision of societal benefits. And you're seeing how that falls out here. We've got 4 main areas we really try to address. You can imagine that with the ocean and all of its issues, this could be an endless endeavor, and it kind of is. So we try to constrain it around climate variability and change, already its own massive topic. Ecosystems, fisheries, and water quality, another massive topic. Coastal hazards, which for us generally these days just is sea level rise and coastal floods. I won't go too much into that today. And then marine operations is another category that we focus on. And we have a sister organization to the north. I probably should have pointed that out on this map. It's a little bit obscured by the orange box, but you can see SCOOS and SCOOS. We're very fortunate in California to have 2 of these regional associations because we are such a large state and we have this large swath of the California current ecosystem that we need to cover. And this picture gives you a sense very quickly of dots and lines on a map of everything we're doing. And I will get into the details on some of these things, but not all of them today. And I will talk a little bit about the high frequency radar. Why do we have that? That measures surface currents. These are radars that are on land all over the coastline, and you need several of them to triangulate on a patch of ocean water, and you get the signal back, and from that signal you can derive the speed of the ocean currents. So this is a really neat tool that has all kinds of applications for search and rescue, oil spill response, you name it. We have these underwater gliders. They're autonomous, remote autonomous vehicles that profile the ocean all the way down to 1,000 meters. That's over 3,000 feet. They're out operationally, which means they're out all the time, 24/7. We've got at least one glider occupying one of these lines that you see here, or multiple of these glider transects that you're seeing here. And we have moorings, we have shore stations doing all kinds of great physics and chemistry so that you always know anytime you want to swim, dive, surf, what are the conditions in the water. And we do some other cool ship-based work as well. And so this is just SCOOS. We have another little picture we've been working on, which gives you a sense of our identity, because we're all a little bit different. It just depends on what are the needs in our region. And you all live in this area, so you probably can appreciate that water quality is a big one for SCOOS. Not every regional association is prioritizing water quality the way we are, but we do prioritize things like harmful algae in the ocean, how good is the water in terms of nutrients, do we need to worry about things like ocean acidification, which is an incredibly important process connected to climate change, whereby the pH of the water is changing in conjunction with the fact that there's a lot more carbon dioxide in the atmosphere. When you put carbon dioxide in the water, which happens naturally when the water and the air exchange, that water gets more acidic, meaning that the partial pressure of CO2 goes up and you actually have more acidic water, just like your carbonated soda, which is also highly acidic. I know I water my plants with my discarded water, fizzy waters all the time, and my plants do not like it. And that's because that water is so acidic. We also have some great projects I'll get into called like the Dynamic Under Keel Clearance Project. And this is a collaboration with a whole lot of entities, including CDIP, that's the wave buoy network, with a little buoy image for you there. These are the high precision wave buoys that are all over the country, but that's our sister program, and they're based here at Scripps, right across the way from me here. And we work really closely with the Marine Exchange on this particular project. So I'm really happy to see Captain Kip Lutet here today, because this is an important partnership where we're really helping to bring the data to the various entities that need to give it to the ships to get them in and out of port. And I'm going to talk a little bit more about that in a minute. So these are our high-level goals. I certainly won't go into all of this. We just wrote a new proposal, so we've got all kinds of language that I could throw at you that might be a little bit boring, but it gives you a very quick overview of how we divide things up. We've got ocean observing, which is everything I just showed you in that picture. Data management, that's what DMAC stands for. This is a huge part of what we do, is not only getting the observations, but doing the quality control, moving them through that pipeline so that we can create products and tools for our users to access those data and look at them and visualize them and use them and interpret them, importantly, interpret them. And at the very end of my talk, I'm going to touch a little bit on the governance and stakeholder engagement. So stakeholder engagement can mean a whole lot of things. We have to work with people like the US Coast Guard, of course, and find out what they need, or say folks at the Marine Exchange and find out what they need. But then we also need to make sure that this information is getting out to people, younger people, people who might not have easy access to this information or even know about it, and give them the sense that not only is this information valuable to every citizen in the country, but also that you're also opening your mind to the kinds of jobs and things that are out there when you see all the stuff that we are engaged in at SCOOS. And so this is a little bit of an overwhelming slide, but what it's meant to show you is that when we say things like stakeholders, we do mean just an incredible number of entities, agencies, groups that we interface with. And this is just not even all of them, but these are the ones we probably communicate with the most in the form of meetings, phone calls, getting out there and just interacting as much as we can with everybody in California, but this is state, this is federal, local, regional, that kind of thing. So we do kind of what one might consider end-to-end, in that we're doing the observing, but then we're also making sure that the data gets used and is useful. So it's not just data, it's also models, and I'll get into that in a minute, but this is another map that we've recently put together that gives you a sense of the things that we are sustaining. And I've already mentioned radar and gliders. We also sustain these monitoring of harmful algae, because I will tell you a little bit more about harmful algae later in the talk and explain why, when we care about such things, And then the automated shore stations. And those are the ones where each of the piers you see highlighted here, we've got sensors that are monitoring temperature, salinity, the chlorophyll, which is the biomass of the algae in the water, and things like that in real time, pushing them, every 4 minutes we're getting a new read, and you can always look at that on our website. And these are the technologies. I've touched on most of these so far that we support. If you were to go to our website, this is what it would look like. You'd see these tiles. You could click on them and look and learn about the technologies and look at the data. And if you were interested and couldn't find those data, you can always get in touch with us if you had any problems. We've also broken it down on our website to what are the variables, what are the parameters that we're measuring? Because what we have found over the years is that there's a lot of different ways to access information, and it depends on how your mind thinks. It depends on what you need. You might think, I would love to find data that come from a glider, and so you access that via the technology. You might be somebody who's developing a model and you want to validate that model, and all you really care about is validating the temperature in your model. So then you might say, I would love to find all of the sea surface temperature that I can find in the region, and then you would access something like this and just go to temperature. And so we've tried to make it really have multiple points of entry for any given data set or any given model or product that we produce. And so speaking of products, there are a lot of them, and we keep expanding these. We're continuing to expand these as we get more funds and find more resources and partners. I'll touch on some of these today, but this is what it looks like when you go to our products page. All right, so returning to the dynamic under-keel clearance project that I mentioned earlier, and this is the one that's in partnership with the Port of LA and Long Beach and the Marine Exchange here, where we are trying to really make that public-private partnership strong and show the validity of what can be done when you bring so many entities together. And so in this case, the big problem, which you may or may not be aware of, is that as these tankers and crude oil carriers are coming into the port, they're getting bigger and bigger because obviously the goal is to maximize how much either fuel you can bring in or how much cargo you can bring into port. And these channels were not designed for that way back in the day. And so we're really pushing the limits of what the port can handle. And so just to illustrate that, when you take some of these big ships and you push a bunch of water at them, and consequently that ship pitches maybe 1 degree in this direction, say, that can result in just about 10 feet of draft change, meaning that that displacement is that big. And if they may only have a few feet margin to work with coming into these channels. So this could be really devastating if one of these ships grounded. And that means that the kind of information that they require is not just the swell and the waves that are hitting the ship, but there's all kinds of information that is specific to how big the ship is, where it's coming from. And so you can put all of that in a model and deliver that information to the ship captains in real time and help them make those decisions. And sometimes this requires lightering offshore, where you would take one larger ship full of oil and take that oil and put it on smaller ships so that those ships can then come into port. And that itself is also a very dangerous thing. And that the same kind of information is required in that case so that you don't have people getting crushed and the problems that can actually ensue when you do this dangerous work. So this has been mostly driven by our partner CDIP here at Scripps, but SCOOS plays a role in helping to deliver some of this information as well. Now moving on to the radar, which I mentioned briefly earlier, we get these surface current measurements. I did say they were great for the search and rescue that the US Coast Guard does, and this is true. So these data go into the models that the Coast Guard would use if they were to go search for a person, or maybe even sometimes it's an entity, but sometimes they're just searching, usually it's a person who may have fallen off of a boat or been swept away by waves, and they need to track them down. And when you have the surface currents, you can more easily figure out where, with more precision, that water might have moved, that patch of water that that person is sitting in. And so we have a picture here of the Coast Guard. That's me in the blue. One of the people in the blue is me. I got to go out and do some work with them. It had nothing to do with search and rescue. This particular project was about monitoring coastline erosion, and we take photos where we can fly kind of low and slow in a Coast Guard helicopter and take photos of the coastline and monitor erosion. So I got to do that one time last year, and it really was just about the highlight of my life so far, getting to get in that helicopter and with such an experienced group. I'd never been in a helicopter before, so if you're ever going to do it, I say that's the team to do it with. But I will mention also oil spill response, because in California, this is a big one. If any of you lived in the Santa Barbara Channel region during the time of the Refugio oil spill, you would know that that was a really big deal and really did affect the marine ecosystem and wildlife and impact them in a very negative way. We have a quote here from Tom Cullen. He's the one who runs the California Office of Spill Prevention and Response. He's a great partner, really sees the value of these data in helping their models, like very similar to the search and rescue, but in this case, you're following patches of oil and you want to know where they're going to go, and you need those predictions. And so these data products are super important for that. But what you might not realize is that they actually are important for water quality as well. This is a slide that is getting at a tool that we have. I didn't show you the tool in this talk, but the tool is called a plume tracker. If you are from down here or spend any time down in the San Diego or US-Mexico border region near Tijuana, you might realize that there is a big pollution problem here, where when there are large rain events, we have, or even not even large rain events, you get a lot of pollution coming out of the Tijuana River Valley into the ocean and the surrounding area. And sometimes you even get pollution that comes out of discharge sites further into Mexico that can flow northward into US waters. And so this is just a big problem. It's that both the US and Mexico are trying to tackle. But a long time ago, SCOOS came on the scene and said, well, we have this radar. Maybe what we can do is project where these plumes of pollution will move when they come out of that point source and disperse along the coastline. Can we help people understand if it's safe to go to the beach, or help somebody who runs, say, a research reserve or an MP or a marine protected area? How can they better protect their habitat and their sensitive species? And so we created a tool that gives you a sense of that exposure. And it basically, you drop like 100 particles simulated in a computer model at that point source, that river source. And then every hour you do that, and you watch over a 3-day period where those particles end up. And that is basically the model that we put out as a tool. And we're trying to expand this to other regions in California. So there's a lot more to that story. I don't have time to go into it today. There's a lot of work being done in this space. And in fact, I just participated last night in a talk for Council Member Moreno down here, thinking about, she runs the districts all around this area near the border. And it was really focused on the pollution problem and things like, how do we measure for viruses? How do we measure for harmful algae and those kinds of things? And I'll talk more about harmful algae in just 1 minute. So ocean acidification, I mentioned that this is something connected to climate change, and it's probably not getting enough of the media attention that it deserves. This is one that, if there's anyone who even doubted climate change, which, we hope there's no more people like that anymore, but this is something that from the get-go has been an expression in the ocean that is a perfectly measurable expression of what it means to put too much carbon dioxide in the atmosphere. Because at some point, what we call the buffering capacity of the ocean just is exceeded. And then that means that you no longer have carbon dioxide being turned into bicarbonate and carbonate and have a nice equilibrium of those 3 carbon species. Instead, what you have is a buildup of that partial pressure of CO2 such that the acidity of the water actually changes, and it's changing on a large scale. And concomitant with those increases, we're also seeing decreases in oxygen. So there's this combined problem that we now refer to as ocean acidification and hypoxia. They tend to go hand in hand, particularly in California, where water that is high in carbon from other parts of the world's ocean has sunk into the bottom of the ocean, and that circulation that's natural on our planet that circulation eventually brings some really, really old water up to the surface here in the Pacific. You might not realize that when you go out and swim on a cold spring day and you're bathing in this cold, newly upwelled water, that's some of the oldest water on the planet. And it can be thousands of years old. And it can be just laden with more acidity than other water, and it can be really low in oxygen. And that's not good for the animals that are going to be bathing in that water constantly. Some fish can move, but a lot of other animals can't. And so we're working hard to measure all of the things related to this. It's a lot of different chemistry that needs to get measured. And we work with engineers here at Scripps that develop pretty cool instruments like the CFOX. This looks at pH and oxygen. That's why it has the name that it has. It can actually calibrate all of those measurements in the water while it's doing its observing. And so we're hoping to get these out at all of the different piers in California and start to really monitor in a very high frequency, so very frequently monitor what is happening and better understand ocean acidification. And then also help our partners at aquafarms who grow shellfish, because the shellfish, as it turns out, they cannot be living in such acidic water because there's not enough carbonate. And carbonate is what they need to create their shells. And when you have really acidic water, that means you also have very little carbonate. And so this is a multifaceted problem, and we're trying to understand all of the ramifications. And so connected to that vision of the fact that I was just telling you we've got old water. So, that's telling you that the water that you see in the Pacific locally is also connected to global circulation. And one way that we can monitor how our little patch of water here in Southern California is connected to the globe is with the gliders that I told you about. And so I wanted to make sure I showed this picture of the way that gliders work, the glider data and what it can tell us. These are some complicated plots. I won't go into them in too much detail, but what I will say is that the upper plot on the left that has all the squiggly colorful lines, that one is showing you all the different glider data combined in one plot with the colorful lines of the different glider lines, showing you what the temperature at 10 meters looks like from 2007 to 2021. And I'll get back to this in a minute. So this long time series, and you can see all the different squiggles, but focus on the black line, because the black line is what's happening at the equator. And that is the Oceanic Nino index. You've probably heard of El Nino. This is connected. It's a way of looking at the El Nino signal. And that normally is going up and down almost in the same kind of way as those colorful lines. However, when you get to 2016, where they split apart and diverge from one another, that's a very important time period. And you'll see if you look in the plot just below it where it's all red, look for the timeline on the left and look for right around 2015, 2016 when it gets really dark red. That's the event that we're still living in some way or other. But this extreme warming event, we call this a marine heat wave, has never really left us. And that marine heat wave is the reason that those lines diverge from one another in the upper plot, because we are finding ourselves in a new kind of pattern or regime where things are just not working the way they used to work. And as you move forward in time, in that time series with those lines, what you see is that sometimes they come back together, which they did in the really large El Nino of 2016, and sometimes they pull apart again, meaning that we're no longer, our little patch of water is no longer as connected to what's happening at the equator as it used to be. There are some changes happening, maybe circulation, maybe something else, and we need to understand what those are because they seem to be connected to a warming climate overall. And so we're trying to understand that, and the glider data give us a beautiful vision of this that we would not have otherwise for the entire basin going out, many hundreds of kilometers offshore. So I told you about the automated shore stations. This is where you can get real-time data. A lot of swimmers like to use this. We know divers use it a lot. And the reason for that is pretty obvious. You can get some great information. Temperature is probably the most important one, but you can also see if the water is clear. That can be a very helpful thing for divers to look at before you go out, say, and do your diving in the Orange County area, if that were something you wanted to do. So now, harmful algae. This is my specialty. This is what's near and dear to my heart. So I will probably spend a lot of time on this. And what I want to say about harmful algae is that we're measuring harmful algae at those same piers where we're doing automated shore station measurements. And we're looking at a lot of things, but let's just focus in on the fact that these pictures in front of you are algae. They're microscopic algae, they're phytoplankton. And they're a subset of the phytoplankton that you would encounter here in our ocean, the base of the food web, the ones that photosynthesize, that really do create the productive environment that we live in the ocean here. And some of them can be bad players, sometimes, not all the time, but sometimes they can become toxic. And we need to watch when those ones are there, when they're growing and when they're becoming more abundant. And then we need to be able to alert people about this, because if those toxins get into the shellfish, for instance, you can become very sick. And so the Department of Public Health for California really does need to monitor that. They need to measure toxins in the shellfish, but they also need to work with partners like us to watch the phytoplankton and do that as frequently as possible. And what about these toxins? I like to show this slide because it gives you a sense of how they rank relative to some of the things you might know more in terms of toxins in venomous, poisonous animals. Saxitoxin is one of the most toxic ones from the ocean, and it's up there with blue-ringed octopus. It's considered a biological warfare agent here in the United States. We keep it in our arsenal just in case we need it. It's that bad. And certain things here in California can produce that, and we monitor them very closely, particularly the State Department of Health does. And then domoic acid. This is another one this is one I've spent a ton of time working on. That's at the bottom. It might not be as bad as some of the others, but when you get domoic acid poisoning, it's akin to, say, a lethal dose from a rattlesnake. So we're not talking about something that's very low risk. It's fairly high risk. And there's a number of reasons why it is high risk. One of those is that for the ecosystem at large, you may have known or seen or heard about marine mammals being affected by these toxins. You may have seen it on the beaches when they strand and they have neurological symptoms and their heads are weaving and bobbing and they're disoriented, and maybe they are even a little too friendly. That can happen too as a result of some of these toxins. And domoic acid is the one we really worry so much about in California because it can get into the shellfish and fish supply, but it also has long-term impacts on sea lions. And sea lions are what we call a sentinel species because they're telling us what's happening out in the ocean. And in the case of domoic acid, they're acquiring a lot of this through what they eat, through anchovies, through sardines, other little critters that they eat that have fed on the phytoplankton. And so now it's getting into their system and it's doing what we call bioaccumulation. So now you've got even more of it, and that load can be fatal, or it can just do long-term brain damage. And those are the two things we're starting to realize are happening with sea lions in California. So I thought I would show you what does the molecule look like. If you love chemistry, you won't be quizzed on this. It's just a good illustration to let you know that in chemical terms, domoic acid is not called an acid because it's acidic. It's called an acid because it's a lot like other amino acids, in particular glutamic acid, an important one that we need. We make it ourselves, meaning that it's a non-essential amino acid. But it's the most prominent neurotransmitter. And it's our main excitatory neurotransmitter. And what's important about that is that domoic acid looks so much like it that it binds to those same parts of the neuron where the neurotransmitters bind to. And then what you end up with is this interference. And so you have breakdown in neural communication, breakdown in memory formation, learning, regulation, all of that. So, glutamic acid can actually be a really good thing. It gives us umami flavor in our food. But if you had Alzheimer's or Parkinson's, what you're experiencing in the case of those diseases is an overabundance of glutamic acid, and therefore this excitotoxicity as a result of that. So domoic acid poisoning and Alzheimer's and Parkinson's all kind of look the same, which is why these animals have the neurological response that they have when they've been exposed to too much domoic acid. So this is a whole other area of research that is really quite fascinating. And I would be remiss if I didn't mention that there's a legacy here in California. If you've ever watched The Birds, the movie by Alfred Hitchcock, it is said and is well accepted now that he was inspired by a domoic acid event, a bloom of a toxic algae called Pseudonychia that produces domoic acid. And this happened in Santa Cruz in the 60s. And it was captured, as you see here on the left, in the Santa Cruz Sentinel, when a bunch of birds were acting pretty wacky because they had been eating lots of fish that were contaminated with domoic acid. And when he saw that and he read that, he realized he could make a connection to the famous book and spin that into a pretty creepy movie about these birds that were falling out of the sky or dive bombing people or acting very strange. And he took it to another level, but there is supposedly a link here in terms of the inspiration. And so I have spent a lot of my career thinking about domoic acid, so much so that I worked with a team for about 15 years to create what you're seeing here on the left. That's how long it took. And this is what is a prediction, like kind of like a weather model that goes out every single day by NOAA. And what it is showing you is what is your risk of encountering domoic acid at a high level in the ocean. So when you see red, that's a high level of a high probability that that water might have a pretty bad or dangerous level of domoic acid in it. If it's more blue or purple, that means that the probability, the risk is low. And we have created this product using all kinds of neat things like satellite data and powerful circulation models. And we put that out with NOAA. So we've basically transitioned it to an operational partner. So NOAA makes this, just like they do weather service models, they do this daily. And this has proved really useful for understanding geographically where things are going. Or if you see a sea lion and it's stranded and you're looking at that patch of water right by the coast and it's blue and you're saying, well, the sea lion couldn't have gotten sick here because it says there's low risk. But then you look at a patch of water just offshore and it's red, you might then say, oh, clearly it was swimming about, 50 to 100 kilometers offshore, and that's probably where this animal acquired the domoic acid. So the marine mammal centers that we work with are really eager to use these sorts of models to better understand where their animals might have gotten sick. And so in order to help them understand that and to help the California Department of Public Health and others understand how their data that are collected sometimes at tiny little sites like a rocky reef or a pier, how does that connect to the bigger picture of what's happening out in the ocean? We created something called the California HAB Bulletin, and this goes out every single month. You can sign up for it. All of the stuff is public. There's a listserv. And if you want to read about it, what it is it's a monthly retrospective that takes all the data from the California Department of Public Health. It takes all the data from every single marine mammal rescue group. They give it to us every month to plot for them and say which animals came into our facility because of domoic acid. And then we take the model and we take everything and we mix it up and say, this is how you should interpret everything that is happening out there right now or in the last month. So it's kind of like this. It looks a little bit different than this, but you get the idea that you've got harmful algal bloom sampling from piers. You have marine mammal suspected cases for domoic acid. The Department of Public Health has its own data of shellfish and other water samples. And then we have the model and we bring it all together and we interpret it so that all of our end users and stakeholders in the community can very quickly assess what are the risks, how does this match up to the thing that I care about. And then one of the reasons people care now is that last year down here in San Diego, but also in your region, there was this historic event of an algae that normally is not dangerous at all. It blooms a lot around here in Southern California. Hopefully you've seen this before. These beautiful blue waves crashing at night during these bioluminescent events. The water will be red or brown by day, but beautiful light shows by night. And normally we would just say, yeah, this is an algae bloom. You could call it a red tide, but it's not really harmful. Until what happened last year, where we had this historic event for 2 months, right around the same time that COVID hit. We had this unusual mortality event where fish, hundreds, thousands of fish, invertebrates like octopus were stranding all over the beaches. dead. They weren't stranding, they were dead. So they were dead because of this bloom. And the bloom had gone on for a long time. And there's a whole story I could go into here. But the upshot of it is that the bloom was around for so long, the water was so warm that oxygen eventually got drained down as that bloom decayed. And another thing was happening that we only started to realize through some forensics is that there was a really bad toxin that it produced that we knew it could have produced, but it's just really never been recorded down here. And so one of the reasons we knew that is this poor little octopus in the lower right, he's bathing in red water that was being brought in from the ocean into Scripps, into a tank, and oxygen was being put into that water. So we knew he eventually died, and we knew it wasn't because of oxygen problems. It was because there was something else in that water that caused that death. And it turns out to be this toxin that is called Yesotoxin. And it's an interesting one that I don't have time to go into today. But we are going to keep monitoring these things. And we just got some extra money to put what are called imaging flow cytobots at all of the piers from Trinidad, that's in Humboldt, all the way down to Scripps Pier down here. And what is that? There are these really neat instruments. We're going to get about 6 any minute now. So we're doing some trainings, which is what these photos are. You can see this is back in Massachusetts where they're creating our new instruments. These are robotic microscopes, and they're very fancy. What they do is they can be underwater, they can be on a pier, they can be in various places that you want to put them. And every 20 minutes, they take a water sample. They take gorgeous images of these teeny tiny microscopic phytoplankton, where ordinarily we would be in a microscope in the lab trying to do that. And then you take machine learning algorithms like facial recognition type software and apply that to those images, and it tells you every 20 minutes what all the phytoplankton are in the water. So now you can tell your manager or public health immediately, this thing is ramping up, or this bug is ramping down. And it's just kind of gonna, I think, blow the lid off of our ability to help people respond to harmful algal blooms in a really, really rapid way. And we're gonna have these on moorings offshore, monitoring what's happening way out offshore. And we're gonna have them on piers as well. And so this is just happening. It's very, very exciting. So we have this vision for the next 5 years where we could have instruments like this everywhere, that we could just pretty much know what's happening in the ocean all the time. And wouldn't that be great? Just have sensors everywhere, have models that are perfect, that predict everything perfectly. So we're trying to get money to do that. It's a big vision, it's a bold vision. It's not one that will be easy to execute anytime soon, but we want to. And I'm gonna skip this slide. It gets in a little bit more into that vision. Just wanna be able to have time for questions. So the last 2 slides are to show you that we have engagement and outreach that involves a lot of different groups, including All2C. And so we're hoping that in the next 5 years, we're going to make some strides in creating webinars, creating instructional information, getting out and really talking to the community more, and helping youngsters out who want to move into ocean science and have an interest in the kinds of stuff that I've been talking about today. So we have a lot of partners. We want to do that. We will be doing various levels of public engagement, including webinars, like I mentioned, career nights, you name it. So, and that there is Jim. He runs, he's the program manager for CDIP. We were doing one of these kinds of outreach activities some years ago together. So I'm going to end there. This is a little slideshow that I will leave it on while we do questions. It's a slideshow from the Red Tide, the bioluminescent event that I mentioned, and it's pretty neat. We got a lot of great pictures, and a lot of these came from citizens who just took pictures. So just the public going out and taking photos and sending them into us. So I will probably leave it here for questions and then stop sharing my screen after we're done with questions.
Awesome. Well, thank you so much. Like I said before, All2C we're thrilled to be partners with Scripps, and this is another example of how this partnership is just beautiful. So thanks again for joining us. And with that, I guess I'll just jump right into questions. I had a question about the algae blooms, because that's something that's been a huge attraction around here. I remember when I was in college, people would go there and take pictures because it's just a surreal sight seeing glowing water at night. And you mentioned this past year has been pretty deadly to the animals. Was this the first time we've seen that toxin that you mentioned affecting so many animals close to shore?
Yeah, so that is a great question, and I didn't give it enough time. So yeah, this toxin, we knew it existed at very low levels, and we'd had one event kind of like this. It was a different dinoflagellate, a different phytoplankton that can also produce this same toxin called Yesotoxin, and this happened up in Sonoma County back in 2011, where a lot of abalone and other animals died. So we knew that it was in the realm of possibility. But in the history of I grew up in Los Angeles, so and have always been in school, as you said, or in some capacity. I've been at a University of California on the coast in California. I've seen these blooms for a long time, particularly in Southern California, and been cued into them. And we have never seen anything like that. I have one colleague who did measure some of this yesotoxin at very low levels in this area once before, but it wasn't associated with animals dying. So we didn't even really believe that that's what was happening at first. We thought this is really just anoxia associated with the fact that this big bloom is decaying. And this foam that you're seeing on the beach, that's another indication you've got algae decaying, and that foam is kind of just a byproduct of all of that. And indeed, I didn't show you the data, but at Scripps Pier, where we had pH and oxygen sensors, oxygen was 0 for an entire week, and pH was 7, which is very low, for an entire week. So that's not conducive to life. However, when we started to get samples and get out, it was very hard during COVID, but get samples all over the beaches and brought them into the lab over the last 6-8 months. We've been measuring those, and we knew that our colleagues in Mexico had measured yesotoxin, because the bloom went from, I should have said this, all the way from Santa Barbara to Baja. So not only was it the longest bloom we'd seen, it was the most geographic extensive bloom. But our colleagues down in Ensenada were measuring yesotoxin, and they were telling me, because I'm pretty close with some of them, and I was saying, okay, this is a problem. So we started to measure it, and we're looking at how we're trying to do the hard work in hindsight of trying to understand, did these animals die because of lack of oxygen? Did they die because of the yesotoxin? Or was it a combination of the two, where sometimes a toxin can just be the coup de grace that actually takes an animal out? This happens with marine mammals and others, where they've got pathology upon pathology, they've got parasite upon parasite, but then there's one thing that just is kind of the last straw. And so you never really know in these circumstances what exactly killed those animals. But it is very interesting that there was so much toxin, more than we've ever measured before. And then on top of that, the toxins seemed to be getting into the air, and surfers were showing signs of respiratory distress. So we partnered with Surfline and Surfrider last year also to put out a survey. We put it out in Spanish and English. We got about 1,000 or more respondents, and 25% of those respondents said that they were having a respiratory distress in response to generally surfing, more than just being at the beach, but surfing and swimming in that water. And that's also something that we've never formally recorded for Southern California.
Wow, it's not good news for the surfers. So it's kind of going off. So David asked a question about, so are these SoCal toxins, example, domaoic acid, the same as those which are infamous with HABs off the Gulf Coast of Florida? So, and kind of tying that in, are we seeing any of this elsewhere, or is it just the Pacific coastline from Santa Barbara to Baja, like you said?
Really good questions. So I'll tackle it. I'll try to tackle it in a logical order for my own brain. Okay, this dinoflagellate. So I'm trying not to use too much of the technical terminology here, but it is important for this conversation. So the type of algae that was happening with this bioluminescent event is called a dinoflagellate, but the species is called Lingulodinium polyedra. The one in Florida is also a dinoflagellate, the one that causes the red tides there, and it's called Karenia brevis. The names of those two sound very different, those two species, but they're actually very related, and their genus for both of them used to be Gonialax way back in the day. But over time, especially with genetic information, these things have diverged, and now taxonomists call them these very different names. Now, the one in Florida has for a long time been known to produce an aerosol, which is kind of like why we weren't totally surprised that this was happening with the surfers. It's just not connected with the one with Lingulodinium, which is the one we see bloom here all the time. And Karenia, however, is super famous for producing a neurotoxin that gets aerosolized. So they actually have to close down beaches when the water is like this in Florida, because people cannot be at the beach. You could die. You could have a very bad asthma attack in response to that. It's called brevetoxin. So they are related, but we do not have Karenia here. It's just a little bit more warmer water species than what you're going to find in California's Pacific. But what I will say about domoic acid and about all of these things is they are generally worldwide phenomena. I just participated in a webinar with a bunch of Russians because Kamchatka, and this was put on by the Russian Academy of Science, because the Kamchatka, a region up there in the, kind of the Arctic, near Arctic adjacent, experienced a really bad bloom of Karenia brevis very recently. And people, you may have seen this in the media, they were blaming it on, all kinds of chemical pollutants from industry, that people couldn't breathe. Swimmers, tourists were having to just flee by the thousands because they couldn't breathe. And it turned out it was the Karenia brevis bloom. And so that is seen worldwide. Domoic acid, also pretty much what we call a cosmopolitan species. Pseudonychia is a genus with many species within it. Some of them are more toxic than others, but you find Pseudonychia all over the world. You find it in Antarctica, you find it up in the Bering Sea. So yeah, domoic acid, however, has not always been a problem in all of the places where you find that diatom, Pseudonychia. We're just seeing it more and more. And very quickly, anecdotally, when I started doing this work, when I was a graduate student at UC Santa Barbara, and this was around, I started in 2000 as a graduate student, and I really wanted to work on this. And my advisor at the time said, he's like, yeah, harmful algae, you get a bloom once every 5-6 years. Why would you do this? That's your whole PhD. You're gonna have one data point from the one bloom that happened in the course of your PhD. But then as it so happened from 2000 forward, we have had a massive Pseudonychia bloom and domoic acid event just about every year from that point forward. I promise I had nothing to do with it. It was a coincidence. But what I learned in that process and what we started to learn over time is how these things have been getting worse. Kind of coincidentally with around 2000, there were some interesting things that happened in the climate around that point, almost like an inflection point. And when I started to do the model that I showed you, the sea harm model with domoic acid, we thought, okay, the level of domoic acid that we should care about right now is probably, let's say 500 nanograms per milliliter. That seemed like a value that was like, that's a bad value. Let's just use that value. And then in 2016, when we had the biggest domoic acid event on record, and went from Santa Barbara to Alaska, we started measuring domoic acid on order 100,000 nanograms per liter, I should say liter. So we went from 500 nanograms per liter to measurements of like 100,000 out in the environment. And nobody could believe that that's where we were, and we're now routinely there. We're routinely seeing levels like that. So what has happened in the environment, we don't know, but things are definitely getting worse.
Those numbers are shocking. So kind of going off that, are we seeing those levels year round or just during that time period? And either way, should this be a huge concern to people that regularly visit the ocean, surf in it? Because you mentioned the respiratory issues they've been having.
Yeah, it really depends on the type of bloom. So when you have a red tide, you're going to see that red, ruddy brown water near shore. A domoic acid event is a little bit less of a thing where, swimming in it's not going to affect you. It's that bioaccumulation thing in the food web that matters more for domoic acid. So it's really more about what we eat. So it is good to pay attention to the advisories. So one of the things we do in the bulletin is put the public health advisories in the bulletin and explain them. And you can also get them off of the California Department of Public Health website. But what they'll tell people on this call, and everyone I talk to, I would like to tell people this, is that while you may be protected from that oyster you want to eat that came from a commercial grower in Humboldt Bay, you're going to be protected because the Department of Public Health is really good at measuring that. But if you just go off to a pier and harvest some mussels, you need to pay attention to those advisories, and you need to pay attention to what's happening in the water, because there may or may not be a sign telling you not to harvest those shellfish. You do need to pay attention to the fact that there could be at different times of year, these things are more prevalent. You asked about that. We're starting to see domoic acid in the water almost year round. It was not like that. Again, I was told not to study this because it would happen every 5 years. Then at least it was only happening in spring and summer for many, many years. Now it does seem like it's becoming much more frequent throughout the year, that the waters in California are just almost always hospitable to this diatom and its growth. The dinoflagellate, however, it's still more infrequent. We see that generally spring and sometimes a little bit in early fall, late summer, early fall. Yeah.
So getting a lot of questions here. Let me try to get through a couple of them. So Mary Watson asked, does Scripps do any animal rescues in the cases of these toxic invasions, or who does? Do you guys partner with anyone that does?
So most of the animals were beyond rescue in the case of, say, that red tide. They were just dead, dead fish, dead invertebrates. In the case of marine mammals, we don't do that at Scripps. Of course, we're working with all of the marine mammal rescue groups. There are many of them in California, and you're probably aware of the ones down here, like at the Pacific Marine Mammal Center, the Marine Mammal Care Center of Los Angeles, and then SeaWorld. Those are some of our big Southern California partners. So they're the ones who are going to be the first responders for any kind of mammal that is on the beach and is alive. When things are dead, it's a little different. Then you've got other groups of fisheries that come in and kind of monitor that, like dead whales and stuff. But at Scripps, we're more concerned with sort of doing the research. Even the animal research that goes on Scripps is more about understanding the physiology that might lead to such a thing happening, but not the ones who are the boots on the ground, get that animal in and get it back out again, or often euthanized, sadly, is the case with a lot of wild animals.
Okay, so just trying to get through all these questions. Fernanda asks, when deploying the CPHO...
That's the CFOX. The CFOX.
Sorry. CFOX probes. I was reading that. I was like, I think there's a way to say this better, but do you take a profile reading or just at one particular depth?
Oh yeah, that's a really good question. We do have things that are profilers. That one, we're not using in that way. We are putting it at a fixed depth. But we can move it around through time and see what are the most appropriate depths. We have a pretty cool instrument. I didn't talk about it at all today. There's an instrument with some people here at Scripps who partner with called a wire walker. You can put anything you want on it. You could put a CFOX or other stuff on there, other payloads, and it ratchets up and down the water column using the wave energy. So depending on the wave energy, it's going to change that frequency of the ratcheting. But if you had a lot of energy, you could be sampling constantly up and down the water column, and it's a really cool thing. So there's ways to do that. We're not focused on that for that project, though.
Got it. So Kip asks, and kind of ties into one of the questions we like to ask as well. Do you have a sense for any change of federal funding levels or focus with the new president and administration?
Good question, Kip. Yeah, so, I think that there is a new emphasis with this new administration, and you've probably all seen that after COVID is the big one. One of the big ones, COVID, and then of course, diversity, equity, inclusion. But I think number 2 on their list is climate change. And so we have actually, I know that my team here feels very relieved now that they can actually say climate change out loud. Because I think for the last 4 years, that was actually pretty problematic. We were really told not to use it. And there were some of us who just ignored that and did it anyway. But now it's a highlight. And so I think in the sense that ocean observing is doing a really, really good job of capturing these long-term time series. And we can talk about the fact that whatever your feelings on climate change are, we need to be monitoring change. We need to understand the attribution of that change. So that could be good, I think, Kip, for us in the ocean observing community. And I would also like to say that, and this is germane to your interests, that the blue economy is also still at the center of these conversations. And so working with the Port of LA and Long Beach and blue economy is still going to be, number 1 or number 2 in our minds. So, yeah. But
Awesome.
I will just say that sometimes when you have a Democratic run Senate, you actually spend less. It's like everyone thinks it's the other way around. We've not been as lucky sometimes when things swapped in that direction, even though you might think that those two things might be correlated.
Fingers crossed. So you mentioned your team. So kind of take us through your background. Like, what made you choose this, and what does a day in the life for you look like in terms of research? You mentioned a lot of webinars, outreach, public outreach.
Oh my gosh, a day in the life. So I will just trek you through yesterday. Let's do that. So yesterday, I had a phone call at 8 a.m. on a UN working group that I'm on that has to do with air-sea interactions and how we're going to plan this out for the UN Decade of the Ocean, which is a really cool thing that's happening. And then right after that, I had a visit with a staffer from the office, Brownlee's office, which is your neck of the woods, to talk about FY22 appropriations and what we think they should be focusing on and what SCOOS does. And then I had another meeting with another staffer in a similar close district. This is Vargas district down here. And then I went into a meeting where I'm leading a project to figure out how do we create a data assembly center for all of the harmful algae data in the country and all of these imaging flow cytobots that people have, so that we'll have this streamlined place where everyone can run all of their machine learning algorithms in one place. And then I had a meeting to work on a proposal for a coastal ocean modeling test bed, where we're trying to figure out how to do nearshore modeling really well down in this area and actually model norovirus content in the ocean for swimmers so that they'll know risk. And then I had another meeting with a staffer and some scientists as well. And then I went on to give a talk last night for Councilmember Moreno. So it's kind of like that, and it's a mixed bag, and every day is a little bit different, just depends on what's happening. We're doing our congressional visits this week, so that's dominating my mind. But my job is very hard to pin down, and it's very exciting for that reason, because it could be anything from that interfacing with stakeholders or Congress or policymakers in California, but it could also just be going out and, oh, I forgot to say, I also went down to the pier to figure out how to deploy an instrument. So I get to do hands-on work, but also keep things at this weird higher level as well. So it's really all over the map.
Sounds like a pretty light workload then, I guess. Just kidding. So you mentioned it's exciting. So one of the things we kind of like to end on every time is, and sometimes we don't have something to end on like this, but optimism. What gives you hope when you look into the future, whether it's your research, your talks with staffers, the webinars you've been doing? What gives you optimism that maybe, that, for lack of a better pun, the tides are changing?
Yeah, that is a nice question. I hope I have a nice answer. You know, what gives me optimism is that there is, because my brain is a little bit stuck right now in that, in sort of that funding and appropriations kind of thing, and what is reading the tea leaves, what do I see happening in the future? So far, I feel very lucky to be in a position that I work in ocean observing, which has incredible bipartisan support. You can imagine that we are working on things that are critical for society, and everybody gets that. And so what I'm hoping is that if we can continue that investment and continue to observe the ocean the way and think about it the way we do weather, which is that these are critical things for our livelihoods, for our communities, and that we can make sure that every single person in this country, citizen or not, has access to these data and to what they need to go out and have a healthy food supply. All of that, I feel like we have the energy and the desire as a nation, as a globe, to really make that happen. I'm seeing tons of investment. What I do worry about sometimes is overexploitation, and over the last 8 years or so, that language has waxed and waned about, well, yes, we should map the ocean, but why do we want to map the ocean? Is it to be as extractive as possible? Sometimes you have to be you have to keep your eyes and ears open about language that's, sheep and wolves clothing, because sometimes people sound like they really want to do something very beneficial for the environment. But the flip side of it, or the hidden agenda, has more to do with, say, increasing or making extraction more efficient. And this doesn't mean I'm anti-extraction for things we need to be self-sufficient as a nation, but just that I really hope we can find a balance, because conservation to me is a big deal. And when we lose species, and we're losing, many a day due to human activity, you can't get those back. And so, I would just leave it there that I'm optimistic, but we need to always remember that we just have this one shot, this one ocean shot and this one earth shot to get this right. And people need to remember that.
That's awesome. Well, we are rounding up the hour. Do you have any final closing thoughts? Anything you want to plug?
Yeah, nothing offhand except keep your eyes and ears open for work that we will be doing with AltaSea, because we will be holding more webinars and doing more activities. So hopefully I'll see some of that again in the future.
Yeah, we got some in the works. So please stay tuned. You can find all the information and a recording of this at altasea-project-blue.org, or just go to our main site, AltaSea, and you'll find it there as well. Dr. Clarissa Anderson, thank you so much for joining us. And I hope everyone has a great weekend.
Yeah, likewise. Thank you so much.